يلا what is a mutation؟ إذا كنا ناقصنا بس رنا لكن يلا. Okay, very good. يلا who can help us؟ يلا what is a mutation? In a few words. Change in DNA sequence. Good يا تحسين. Is it random ولا in a specific place? It is random. طيب let's say, do we know when it's going to happen ولا it happens suddenly? Happens suddenly. Okay, very good. So it could be spontaneous or induced. طب what are the factors that can cause a mutation? What are the factors that can cause a mutation? Yalla, open the microphones and answer.
Is this being exposed to ultraviolet rays?
DNA exposed to?
مثل exposed to X-rays.
X-ray radiation or to chemicals?
Yes.
So is then radiation or chemicals. Okay, so radiation or chemicals. So it could be sometimes beneficial, like in the case of bacterial resistance, and in most of the cases it is what? It is harmful. In most of the cases it is harmful. طيب. Okay. طيب. Who can tell me types of mutation? So mutation can lead to diversity, as you all know, producing new alleles. فإذا type of mutations, we have the genetic, we have a chromosomal. Let's talk about chromosomal first. It can be either an increase or decrease in the total number of chromosomes, or it can, let's say, losing a small piece of the chromosome or duplicating another small piece. طب, what about now gene mutation? What about gene mutation? Types of gene mutations.
It says عندنا silent mutation.
Silent. Why you call it silent يا مريم?
لأنه إنه مثل ما بيبين عنا بال.
So it has no effect on the phenotype.
Yes.
So no change in the amino acid produced.
Yes.
Okay. And?
missense.
Missense, which means what?
إنه بيبين فرق.
Change of one amino acid, a substitution that causes a change of one amino acid. Okay. And we have the third type, which is what?
says ماني ذاكرته يعني اسمه بس إنه هو ال change لكذا amino acid.
Okay, so it's called the frame shift.
Yes.
Which is either addition or substitution. Either addition or substitution.
Yes.
Okay, so this can change the whole sequence or produce a smaller protein. Produce a smaller protein. Okay. Type. Example of the gene mutation, we have the multiple alleles in the blood group A, B, and O. It differs in the substance that is, let's say, shown on the surface of the red blood cell. N-acetylgalactosamine in the case of A, galactose in the case of B, and none of them in the case of O. Okay, document 3. So this can lead to polymorphism. Okay, mutation can lead to polymorphism, causing diversity, like in the case of HLA proteins, or what we call as they are the MHC, which are coded by 6 genes. Okay, and no one has the same, let's say, combination of MHC due to what? Due to the high numbers of alleles of these genes. So each one of us will take one allele by random. So that's why this will be, let's say, impossible for two humans to have exactly the same MHC, except in the identical twins. or mutation can lead to a disease, like for example sickle cell anemia, thalassemia, and so on, caused by a change in one amino acid in the hemoglobin. This was due to change of a nucleotide. Change of a nucleotide. Then we moved on to the detection of a genetic polymorphism. طيب, other than Maryam, who can tell us what are restriction enzymes? Other than Maryam. Restriction enzymes. يلا جنى, صبحية, عبد الله, قاسم, محمد, تحسين. What are restriction enzymes? Yalla, who can help us with this? Restriction enzymes, what are they? DNA cutters. Good ya Tahsin. DNA cutters. طيب, صبحية, do they cut randomly in any place of the DNA? Well, they have a specific sequence? صبحية, do they cut any place, anywhere?
لا أستاذ specific.
Specific. What do we call these? Restriction site.
Yes.
فإذا each restriction enzyme should recognize a restriction site in order to cut at this site. طب what happens يا صبحية if you change one nucleotide in this site? If we change one nucleotide in this site, what happens?
أستاذ بصير ما بيعمل cut.
فإذا it cannot cut anymore because it will not recognize this site. Okay, so change in one amino acid in this place, so it will not be able to cut anymore. طيب, how we can detect this? How we can detect this? We can detect it, okay, using the technique or the method called gel electrophoresis. Okay, the technique called gel electrophoresis. So in the gel electrophoresis, we put the DNA fragments in a gel, and then we put an electric current. The electric current will push away the fragments. They move according to their size. According to their size, the smaller piece will move farther away from the beginning. Okay, that's how we can know, ya Subhiyya, if this allele was cut or not. Meaning, if this allele has a mutation or not when you compare it with the normal. This what leads us to discuss the RFLPs, restriction fragment length polymorphism. Okay, this is comparing each human specific parts of the DNA that he doesn't share with anyone else. Okay, specific parts of the DNA that it doesn't share with anyone else. So this what we can how we can identify the cells and the blood drops of any person by comparing it to the DNA of the database that we have, for example, in the computer. So we can identify because no one shares the same RFLPs, or if you want to say DNA map or restriction map, with anyone else. Can we benefit from this in real life? We say yes. The first application is a genetic identity of individuals. Genetic identity of individuals we can know. For example, first with the FISH technique, fluorescence in situ hybridization. Fluorescence يعني giving radiation, in situ in its place, hybridization because there is complementarity. Okay, Meaning, if I want to know if someone has this allele in his gene or not, I get a piece of DNA that is complementary to this part. It's called a probe. Then I attach to it fluorescent tag, fluorescent molecule. I insert it inside his nucleus of his cell. And then if this part attach, we know that he has this allele. If it doesn't attach, so he doesn't have this allele. For example, you're searching for a disease allele, okay, in the cells of a fetus before the fetus is born. Also, the application can be the DNA fingerprint ch3 method, how we do a DNA map for each human, and then we can compare it to identify whose DNA is this. We can use it in the paternity testing and the criminal investigation, dead bodies identification. Okay, any question about this part? Any question about this part? Let me see. Okay, Tahsin said it's clear. طيب, if you find it clear, please go to question 7 on page 106. Okay, question 7 on page 106.
Says احنا عنا ايه 101.
ما مشكلة. فإذا page 101 in your book. No problem. Thank you يا عبد الله. Page 101 in your book. Okay, yalla, who wants to read? Yalla, who wants to read? جنى، صبحية، قاسم، محمد، عبد الله، تحسين.
أستاذ بقرا؟
لا، ويت يا مريم.
أستاذ أنا بقرا إذا بدك.
تفضلي يا جنى.
Okay. Pascal, 4 years old, went through 2 dysuria attacks, difficulty in urination, in 6 months interval.
يعني imagine a 4 years old boy has a problem in urination. He cannot enter the bathroom. Okay?
The first attack has finished by spontaneous expulsion through urine of small crystals, salt particles with 90% of hydroxyadenine.
Hydroxyadenine.
The second
okay.
hydroxyadenine. The second attack necessitated a surgical intervention, take out of 26 crystals.
فإذا you find in his urine crystals of hydroxyadenine. And the first time he expelled them in the urine. In the second one, he needed a surgical intervention, يعني surgical procedure to take out 26 crystals found in his urine, blocking the way so no urine is coming out. Okay.
This child was asked for several clinical analyses. Document 1 summarizes the obtained results.
طيب, look at the results يا جنى and tell me what you can see. Adenine in the urine of Pascal. How much? The amount?
40.
And the normal individual should be?
1.5.
1.5, يعني 25 times, around the 25 times higher. This is one. What about hydroxyadenine?
It's a high amount
high
in Pascal.
amounts in Pascal, while in normal individual
Traces.
traces. Okay. After ya Jana, comparing the results, derive a conclusion concerning the biochemical disorder of Pascal. Give me first ya Jana comparison.
And know the adenine in the urine of in Pascal during the 24 hours
Okay.
is 40, which is
40 what?
high 40
milligram.
موافق. هدي milligram. Okay, فكرت أيونات غريبة. Okay.
يلا.
Which is 40 milligram. That is higher amount from the normal individual.
Okay.
Which is 1.5 milligram.
طيب. In addition, hydroxyadenine.
It should be traces in the normal. Often in Pascal, it's high amount.
Yes.
Higher than the normal individual. That is traces.
Okay, higher than the normal individual who have traces. So, what could be the conclusion here, you think?
The high amount of adenine. No.
لا, give me conclusion about the problem of Pascal.
You have extra crystals.
لا هلا in normal people الأدينين amount in the urine should be 1.5.
يعني عنده extra.
فإذا he has an excess adenine, so he has a problem in, let's say, eliminating adenine. يعني his body cannot decrease the amount of adenine from 40 to 1.5. Right?
يعني معلقة على الأدينين وهيدروكس.
But hydroxyadenine is like a kind of a product of adenine. ونحن يا جنى we know the adenine صح؟ ما we know the adenine؟
لا.
بلا كيف لا؟ DNA is made of what?
2 sequences.
Okay, what are the bases of the DNA?
هن الأدينين يعني.
شو هن البيزز؟ جنى name the bases of DNA.
ماني حافظتهم.
يه؟
ال A قصدك
لك
والC G.
هول هن في غيبهم ليش؟
طيب شو دخل الأدينين فيها؟
ال A هو symbol of what؟
للأدينين؟
لكن
ماني حافظة اسمائهم.
A is for adenine, T thymine, C cytosine, G guanine. فإذا his body contains adenine, but he has a problem in eliminating this adenine. He has a problem in eliminating this adenine. So, read the answer please, ya Jana.
In case of Pascal, the amount of adenine emitted in the urine in 24 hours is 40 milligram, which is much higher by approximately 25 times than that in the normal individual, that is 1.5 milligram. Also, the amount of hydroxyadenine in the normal individual is not detectable.
Okay.
While it is high in Pascal's case, so Pascal's disorder is due to a problem in the elimination or decomposition of adenine.
فإذا Pascal disorder is due to a problem in the elimination or decomposition of adenine. Or decomposition of adenine. Okay?
Yes.
طيب. هلا in the organism. يلا يا جنى, let's continue.
In the organism, adenine is normally eliminated and decomposed according to the following reactions.
Okay.
بقرا هول الثلاثة.
Let's look at the figure. Yes. Adenine, some of it remains as adenine, 1.5 milligram in the urine. This is the normal amount. Okay, the rest 10% of it will be converted to hydroxyadenine, and 90% by the enzyme APRT converted adenosine monophosphate and used again.
هذا بالنورمال يعني؟
ايه يعني normal الادينين عندك only 1.5 milligram comes out in the urine. The remaining amount في 10% بتصير hydroxyadenine يعني very few amount, وال 90% يعني majority of it converted by APRT into what? Into adenosine monophosphate AMP. So indicate يا جنى using document 2 and the obtained result of Pascal in document 1 the fate of adenine in Pascal case and conclude the cause of crystal appearance. Look at this figure and look at his results. What you can deduce here? What can you conclude?
إنه عنده منهم أصلا نفس الطريقة تاع النورمال، إنه مش 10% و 90%.
اوكي، فإذا أيو إذا بدك pathway of these three he has a problem in it. أي وحدة اللي فيها البروبلم؟
بالهيدروكسي أدينين؟
لا، هو عم يطلع عنده هيدروكسي أدينين بكميات عالية.
يعني منها إنه في اختلاف منها 10% تكون أعلى.
طيب شو اللي ما عم يصير عنده؟ شو اللي ما عم يتحول؟
ما عم بيتحول عنده آخر وحدة APRT.
فإذا no production of AMP. فممكن تكون البروبلم in the enzyme APRT. صح؟
Yes.
إنه الانزيم APRT does not convert 90% of the adenine into AMP, adenosine monophosphate. So now most of the adenine is converted to hydroxyadenine and the remaining high amount remain as it is. That's why in the urine of Pascal you see 40 milligram adenine or you see high amount of hydroxyadenine. صح؟
Yes.
Okay. So you read the answer, please.
Yes. Document 1 shows that Pascal's disorder is due to a problem in the elimination or decomposition of adenine.
Okay.
Document 2 shows that in the normal individual, part of adenine is eliminated in the urine, 1.5 milligram, and the remaining amount is decomposed into hydroxyadenine, 10% by certain enzymes and adenosine monophosphate, 90% by APRT enzyme.
So
based on the above information in case of Pascal, adenine is either eliminated in the urine or decomposed into hydroxyadenine. So crystal's appearance in case of Pascal is due to the non-functioning of the enzyme APRT and hence the production or accumulation of a huge amounts of hydroxyadenine.
okay, so the enzyme is not working, so most of the adenine either remain as it is as a 40 milligram or converted to hydroxyadenine or converted to a hydroxy.
يعني كله بسبب الانزيم.
Exactly. Enzyme here is not working. For example نحن لأنه هيدي conclusion. Okay, conclusion إنه كل الأدينين, all the adenine is either eliminated as adenine أو as hydroxyadenine.
يعني صار في mutation بالأصل مشان هيك ما عم تقدر تشتغل الانزيم.
هذا اللي أنت عم تستعجليه هلا. عم تستعجلي. يعني هلا أنت عم تتنبئي بالريزلت أو بالكوز before we do any testing. صح؟
اوكي.
طيب. Read the question 3 please.
Okay, formulate a hypothesis concerning the origin of the biochemical disorder of Pascal's body.
هلا أعطيني الهايبوثيسيس اللي أنت قلتيها. شو سبب الديزوردر عند باسكال؟
The disorder of Pascal body is due to mutation.
To the mutation of what?
of the enzyme.
ها هلا بنقول of the gene producing the enzyme APRT.
الجين اللي عم
لك
ينشئ
لأنه الميوتيشن بتكون وين عادة بالجين.
Yes.
فإذا read the hypothesis please number 3.
Hypothesis: Pascal's abnormality is due to the deficiency of APRT or presence of an abnormal inactive APRT enzyme.
Exactly. فإذا يا إما lack of APRT أو abnormal inactive APRT. صح؟
Yes.
طيب until now guys, okay stay with me يا جنى. Until now, is it clear for everyone?
Clear.
For everyone. Okay. Do you see anything from your, let's say, acquired knowledge or everything in front of you in the documents? فإذا everything in the documents until now. So that's why we need the training on how to deal with the documents. Okay? Just the training. يعني solving more and more questions. Solving more and more questions. فإذا أنتوا you get used to the type of the documents you see on how to deal with them, how to read them, how to extract information from them, and so on. Okay? Okay? طيب. Let us continue يا جنى. يلا.
Okay. And experimentally, measurements took place to verify the hypothesis formulated.
Okay, we want to check if your hypothesis is correct or not. So what we did, look at document 3. Now, what do we have?
And a percentage of active
APRT enzyme. In Pascal is how much?
0.
0. In the normal people?
100.
100. فإذا now you are 100% sure that the enzyme in Pascal is not working. The enzyme in Pascal is not working. Okay?
أستاذ.
Yes.
طب بركي الميوتيشن موجودة بالإنزيم اللي بده يعمله.
بده يعمل مين؟
يعني ما الإنزيم بالعادة بيعمل قط لشيء معين؟
هيديك اسمه restriction enzyme، هيديك نحن ما عندنا اياه normally. ما نحن we took it from bacteria يا جنى restriction enzyme.
اوكي.
اوكي؟ فإذا هون نحن we are checking إنه the gene in the body of this person is not working. Okay, the gene in the body of this person is not working. Okay? فإذا now you make sure about that this gene now is not working in Pascal لأنه percentage of activity in Pascal 0. Percentage of activity in normal people is how much? 100. طيب, now compare the obtained result. What can you conclude concerning the validity of the hypothesis? Is the hypothesis that you put was correct?
Yes.
Yes. طيب, when we compare, أكيد I will use the numbers. So the percent of active APRT in case of Pascal is 0, while it is 100 in the normal individual, much higher. So Pascal disorder is due to an abnormality in the APRT enzyme, which, what, I missed the remaining of the answer. Validate the previously formulated individual. guys بس give me بس 1 minute please. Okay, بس 1 minute. Okay, sorry guys بس طلعت شوي. طيب جانا can we continue please?
Yes, أكيد. A study is done to show the genetic origin of this problem. The following sequence represent a fragment of the coding strand of the gene responsible for the synthesis of APRT enzyme in the normal and in the affected person.
فإذا now we are comparing the allele from the normal person with an allele from an affected individual. Okay, let's compare now the normal individual with the affected individual. طيب, let's look at these two. Okay, are you still with me? بعدكم معي؟ الو؟ الو بعدكم معي؟ سوري بس لأنه قطعت الكهرباء شوي. طيب جنى, let's compare please جنى. Let's compare please. Between normal individual and affected individual. Okay. جنى سمعتيني؟
استاذ اذا بدك كمل انا.
طيب يلا يا مريم كملي نشوف. بس لأن يمكن بكي جنى قطعت عندها كهرباء أو صار عندها شيء كمان. يلا who can continue please.
أستاذ كنت عم بحكي بس شكلك ما كنت عم تسمعني.
ممكن والله ممكن لأنه قطع كمان عندي وجه اشتراك مرة ثانية بس هلا شكله إن شاء الله الحمد لله مشي الحال. جانا خلينا نكمل هالبارت. يلا بقي معنا 6 minutes.
أستاذ عم لك الفورث سيكونس إنه الأي صارت تي.
فإذا شو نوع عندي هون أنا شو اسمه؟ نوع الميوتيشن؟ substitution. أوكي. نوع الميوتيشن هو substitution.
بس السبستيوشن بتكون لما بيصير
ما شو يعني كلمة
إنه شو
substitution يا جنى؟ كلمة substitution شو يعني شو معناتها؟
إنه الشي بيتنقص
شي بشو؟
يعني بيتبدل أو شي يعني.
بيتبدل تماما. فإذا the sequence of DNA is the same in the normal and affected individual except in the first nucleotide of the fourth triplet where A is replaced by T in the affected one. دائما لما بدكم تحكوا عن الميوتيشن please بدكم تحددوا اللوكيشن exactly where it happening. يعني don't say yes يا تحسين سامعيني؟
سمعتك.
اوكي ما بعرف بحكي عن تحسين ما عم يطلع الصوت. اوكي هلا do not say بس انه mutation by substitution A by T وخلاص لا. You have to say exactly where it happens. يعني here in the fourth triplet. Okay, in the fourth triplet, the first nucleotide A is replaced by T. Okay, the first nucleotide A is replaced by T. تمام؟
Yes.
طيب. Number 6. يلا يا جنى the last one.
Give the amino acid sequence encoded by each of the given fragments. Derive a conclusion.
Okay, Jana, this is the coding ولا non-coding strand?
The coding.
Coding, it's in the given. قالوا لنا انه هيدي الcoding strand. Okay, فإذا now from this one أنا I make messenger RNA complementary يعني ال AU, UGG, GGC and so on in both. بعدين بعمل amino acid sequence. بعدين I derive a conclusion.
Yes.
Okay.
أنا بجيب الأمينو أسيد من أعطيني هون تيبل.
منو عاطيني شو؟
ما في تيبل هون، من وين بجيب الأمينو أسيد؟
هو عادة بيعطيك التيبل يا عبد الله، عادة بيعطيك التيبل. اوكي؟ عادة بيعطيك التيبل. طيب جانا can we read the answer please؟
Yes. Normal individual. ضروري يقولون هولي؟
ايه يلا ثريونين برولين لوسين ارجينين لايسين الانين.
Yes. Knowing that in messenger RNA, same base sequence as the DNA coding strand, except that T is replaced by U.
Okay. Continue.
Affected individual. اللي هن هولي يعني. Therefore, the biochemical disorder in Pascal's case is due to a substitution mutation. A nonsense mutation leads to the appearance of a stop codon before its normal position in the DNA gene that encodes the production of APRT enzyme, causing the synthesis of modified, abnormal, and thus inactive non-functional APRT enzyme protein.
You see بالاffected individual صار عندي mutation عمل لي stop codon. الستوب يعني translation does not continue. فوقف فطلع very small protein inactive non-functional APRT enzyme leading to this problem. Okay, leading to this problem. Okay, clear?
Yes.
اتفقنا كلياتنا ان شاء الله؟ Any question? Any question? حدا سمع شيء عن الفترة اللي جاية شو حيصير؟
وصل العدد 3500 فبحس حيكون في دروب.
حيكون في يمكن إقفال يمكن ما هيك والله أعلم.
أنا هيك بحس ما سمعت شيء بس إنه لأنه كتير زاد العدد أكتر بكتير من قبل.
طيب على كل الأحوال نحن خلونا ناخذ number 8 as a homework. اوكي و if you want to send it to me so I can correct it for you in the مثلا بهاليومين اللي جايين بيكون كتير منيح. اوكي بدي اياكم تجربوا فيه وتجربوا تحلوا.
شو هو 8
8 number 8.
ايه اوكي
اوكي
yes
يلا thank you for your attendance. السلام عليكم.
وعليكم السلام.